Good morning, everyone. It's Friday. It's 8:30. You know what we do. We're celebrating vision research that day. And today we have, it's no exception. We have fantastic researchers from Alabama. And the topic will be related to aging to some degree, right? As we age, except students never age, but as we other people, normal people age, we're losing our dark adaptation over time. So that process is very important, and it could be also indication of the disease. So today we have really someone who contributes most in this area to tell us a little bit more specifics. And with that, I would like to move to Cezary for formal introduction.
Good morning, everybody. My name is Cezary Rydz, and I am a postdoctoral researcher at the Skowrońska Chemistry Lab here at Gavin Herbert Eye Institute at the University of California, Irvine. It is my great honor to welcome you to today's lecture and present our today's speaker, Dr. Cynthia Owsley. Dr. Owsley is a professor, Nathan Miles Endowed Chair of Ophthalmology, and the Director of Clinical Research Unit at the University of Alabama at Birmingham. Dr. Owsley is a Phi Beta Kappa graduate of Wheaton College in Massachusetts and completed her Ph.D. in experimental psychology, specializing in visual perception at Cornell University, where she was a National Science Foundation predoctoral fellow. Her postdoctoral training was at Northwestern University in Vision and Aging. She then completed the Master of Sciences in Public Health and Epidemiology at the University of Alabama, Birmingham's School of Public Health. Dr. Owsley has been continuously funded by the National Institutes of Health since 1983. Dr. Owsley's research focuses on the impact of aging on vision. Her research efforts address why some older adults in normal macular health transition to age-related macular degeneration. Dr. Owsley's research also addresses the relationship between vision and driving and strategies that can improve routine eye care utilization among populations at risk for eye disease and vision impairment. She is particularly interested in the transition of research findings into public policy in these arenas. Throughout her fruitful career, Dr. Owsley published multiple papers in prestigious journals on vision and received multiple awards. She is the recipient of the Glenn Fry Award from the American Optometric Foundation and the Bartimaeus Award from the Detroit Institute of Ophthalmology. She is a Gold Fellow of the Association for Research in Vision and Ophthalmology, a member of the Scientific Advisory Committee at Research to Prevent Blindness, and a member of the Board of Directors of Prevent Blindness. Without further ado, please welcome Dr. Cynthia Owsley.
Thank you very much for inviting me to speak in your lecture series, and thank you very much for that very kind introduction. Today, I'm going to be actually giving you a bit of history on our work on Rod-Mediated dark adaptation as it's relevant to aging and AMD. This saga has gone on for a very long time, approximately 25 years, and it's been a very interesting part of my career, and I hope to summarize it for you today. These are my financial disclosures.
Dr. Owsley, can you just share your screen?
Sorry?
Can you share your
slides?
Am I muted?
No, you're not sharing the slides.
Oh, okay. Well, let me start over. Hold on. They were working before. Let's see. Can you see them now?
Not yet.
Wait, hold on again. Okay, let me go back.
Yes. Now
Can you see them now?
Perfect.
Yes.
Okay, super. So these are my financial disclosures. And in studying AMD, there's really two large areas how you can approach this topic. And the question that we asked ourselves years ago was, what should we really focus on? And so there's one topic is AMD prevention, where we focus on biological events that are basically laying the foundation for later photoreceptor death. And this is what we focus on. Although most of the research in AMD, as we look not just at the human studies, but also the basic research studies, they're focused on arresting progression to end-stage disease. And this, of course, is a very important part of the research. We all know the fantastic achievements in preventing CNV with the anti-VEGF treatments, but also much of the field right now is focusing on to arrest progression to geographic atrophy. And there's many trials, as I'm sure you're all hearing about, that are starting. They're either ongoing, and it's a very exciting phase. However, we choose here at UAB to focus on AMD prevention because it's really a neglected part of the disease. So my own background is actually not so much in biology, but my background is in experimental psychology. And so my specialty area is actually psychophysical methods in using these to study older adults. And one of my major goals throughout my career is to basically, when we're thinking about AMD and particularly studying the early phases of the disease, is developing functional endpoints that can be used in trials. But it's important to keep in mind that really the best functional endpoints are those that are related to structure. And so I'm going to describe our all-star studies to you, where we're trying to identify functional endpoints, but a major part of the research program that I'm involved in collaboration with Christine Curcio and also others at Tahini like Vas Sada and Joel Hugh is to try to relate the functional endpoints to structure. So right there in Orange County, I've been attending first the Beckman, now the Ryan meeting. And back in 2019, we actually had a thematic group that was focused on multimodal imaging and psychophysics. So this is the last time we really focused on psychophysics, but our work was very exciting at that time. And basically, we made several recommendations as a group. First of all, structure. We recommended that structure function assays should be selected that are motivated by the pathobiology of AMD, rather than say just picking visual acuity, because that's what ophthalmology always uses. We also acknowledge that different phases of AMD natural history may actually call for different structure function assays as endpoints. What's happening at end-stage disease is not necessarily what's happening as the disease is developing. And it's also important to realize that when we identify endpoints, when we identify structural changes or functional changes, they're not always, they can't always be used as endpoints, because the endpoints have to be repeatable. They have to have large effect sizes and so on. So that's really the translational challenge in AMD research today from a clinical standpoint. So as we all know, there's treatment options for AMD that are FDA approved. There's treatment for arresting the progression of choroidal neovascularization. There's also treatment for slowing progression using nutritional supplements focused on intermediate AMD. But there's really no treatments for arresting early AMD progression. There's no treatments for preventing AMD in those who are at risk, for example, family history, those who are smokers, those who have other characteristics that elevate risk. So really the barriers to developing these types of treatments are, we really have a poor understanding of the transition from aging to AMD. And as I've already pointed out, there's no established functional endpoints for trials that are targeted at early disease. I get calls from pharmaceutical companies all the time as they're developing earlier treatments, and they're really searching for an endpoint that they can use in a trial. So there were really 3 stimulating events that brought me to this field, and I'm going to summarize these briefly for you. of course, Christine Curcio, who I mentioned. I'll talk about her findings. Unfortunately, Sam Jacobson very sadly passed away recently, but I had a collaboration with Sam during the 1990s that I'll mention. And also we were stimulated by observations from Alan Bird's group in London on early AMD. So it's important for us to realize that even though we think of AMD as a cone disease and it involves the fovea, which is exclusively cones, if we look at the macula, rods actually dominate the macula, as shown in Christine Curcio's work. And rod loss, not cone loss, is really characteristic of both aging and early AMD. And during aging, the rods decrease by 30% near the fovea, but the cones are relatively protected. And so during AMD, rod loss typically exceeds cone loss. And in end-stage AMD, the surviving photoreceptors are largely cones, but however, in end-stage disease, they also die and you can lose foveal vision. So back in the 1990s, Sam Jacobson and I found out that we both had the same modified Humphrey visual perimeter that Fred Fitzke in London made for us. And the reason this perimeter was useful is it could do scotopic sensitivity testing as well as the usual photopic testing that Humphrey field analyzers normally do. And so we did a study focused on older adults and also those in early AMD. And what we found is that photopic sensitivity deficits can occur, but they are much less prevalent and much less severe than rod-mediated deficits, which is pictured on the left. And so this basically confirmed for us, it was the first, I think, psychophysical evidence for rod-mediated functional vulnerability in aging and AMD. And you have to keep in mind, these scotopic sensitivity measurements were done under steady state conditions. In other words, the participants sat in the dark for about 45 minutes so that they, in fact, we could measure scotopic sensitivity. So from there, we also noticed there was an article by Alan Bird's group that studied dark adaptation in about 10 patients. And these people, like I mentioned, were in early AMD, and I've highlighted some of the significant findings. They said the kinetics of dark adaptation were abnormal in all 10 patients in whom reliable measurements were possible. The findings suggested that visual symptoms reflect abnormality of both scotopic sensitivity and also the time course of dark adaptation in patients with early AMD. And so we were really intrigued by that. I think most of you are probably aware, but just to review what dark adaptation is, it's a psychophysical test. Basically, over here, you get a very strong bleaching light, and then you basically measure the recovery of sensitivity in the dark following the bright light exposure. And as you can see, it's very similar to, say, walking into a dark theater on a very sunny day, and it takes you a while to adjust or adapt to the darkness. So as you can see here, the cone recovery, which I'm sure most of you know, is very rapid. However, it takes longer after a photo bleach for the rods to recover. And basically the slope of the second component of rod-mediated dark adaptation approximates the rate of rhodopsin regeneration. So here in red here is the second component, and this could actually be used to measure the rate of dark adaptation. So in Alan Bird's work, here on top with the large open and filled circles are two people with early AMD, and down here are normal people, and you can see that the recovery of sensitivity is much slower in those with AMD. And this kind of stimulated us to work further in this area to kind of explore some of Bird's speculations. So he basically speculated that this phenomenon was due to a thickening of Brooks membrane containing high level of lipids. In some eyes, this was a diffuse layer rather than others. It was discrete deposits, for example, drusen. And he hypothesized that it's a barrier to metabolic exchange, basically creating a localized shortage of vitamin A. And so this really intrigued us. And so basically, it set us working with these and reading about these scientists. It really propelled us forward to study rod-mediated dark adaptation in early AMD. So basically, I started working with my graduate student, Greg Jackson. And one of the very central things we did, okay, keep in mind in AMD research at this time, so there were a lot of epidemiological studies being done, and people created AMD fundus grading systems so you could compare across studies, which I think is important because different clinicians, some clinicians might call it early, some might call it intermediate, use those terms differently. But we actually started when we studied dark adaptation, when we studied people in aging and AMD, we use these AMD fundus grading systems to document not only the presence of disease, but the severity of it so that we could study dark adaptation. So we basically confirmed the hypothesis that rod-mediated dark adaptation is slowed in AMD. These three are AMD subjects. Here's a normal subject, and as you can see, it's delayed. And we thought these data were consistent with this hypothesis that AMD changes in Brooks membrane and RPE complex may be causing an insufficient supply of 11-cis retinal to the rod outer segments, therefore slowing the visual cycle. Basically, we went on from there. We were also interested in cone-mediated dark adaptation, and there were varying reports on this in the literature. And what we found is that we found very little impact in early AMD on cone-mediated dark adaptation. Here's examples of the rod recovery. You can see the delays, but the cone recovery in aging and AMD greatly overlapping. Not all studies agree on this. Part of it may be having to do with studying disease severity using fundus grading systems and so on. But we basically thought that the cone effects were much more modest than what we were seeing with respect to rods. And why would this be? Well, we thought about this a little bit, and we realized that cones have a second visual cycle. They're just not focused. They're just not focused on the only visual cycle that rods have. And so we thought, well, maybe cones can bail out and have this alternative source. So basically, we were thinking that the lipid-rich lesions, drusen and also reticular pseudodrusen, which more recently Rick Spade and Christine Curcio have called SDD, subretinal drusenoid deposits. These lesions likely hamper metabolic exchange, and it creates a diffusion barrier that impedes translocation of lipoproteins to the photoreceptors. And so basically, the reduction of vitamin A reduces 11-cis retinol available, and the functional implication in everyday life is that dark adaptation slows. So about this time, Greg, Christine, and I decided to write kind of a position piece, short position piece in investigative ophthalmology, that perhaps we need to rethink the approach of exactly how we go about studying AMD and focusing more on prevention. And we thought, well, if we could spare the rods, if we could save the rods, well, maybe we could save the cones in later disease. And so we basically argued for a focus on studying earlier disease and the mechanisms underlying the demise of rod photoreceptors rather than just focusing on why cones die. This stimulated a lot of research from not just our group, but other groups looking at early functional biomarkers of AMD and interest in developing endpoints and outcomes involving rod vision that could be used in AMD trials. This also led us to develop an apparatus to measure rod-mediated dark adaptation. At the time, there was only the old Goldman Weakers that was available, which was not computerized. But my student went ahead and worked through the SBIR process and actually developed an apparatus that is on sale today, and researchers can purchase this and study dark adaptation. So just as an aside, I want to tell you about briefly a proof of concept study. What we're wondering is that if we could push a very high dose of vitamin A to the photoreceptors, could we actually speed up dark adaptation in older adults and those with early AMD? And so we actually did a randomized double masked placebo-controlled experiment where what we did was we had a baseline visit. We measured dark adaptation at this time, and we then randomized people to 50,000 international units of retinol versus a placebo. And this is a very high dose. You can only be on this briefly about 30 days. And we had medical monitors monitoring patients. And then what we did was, after the 30 days were up, we looked to see if we could speed up dark adaptation. And we didn't do this because we think high dose vitamin A is a treatment for AMD, but we wanted to see if we could move the parameter of the rate of rod-mediated dark adaptation. And basically, we did. We basically found that the high-dose vitamin A increased the speed of rod-mediated dark adaptation, but it didn't impact the cone parameters. And what was, excuse me, what was really interesting is that our patient-reported outcome measure, this is a subjective report, people didn't know what they were on, but those patients who had greater increases in their rod slope reported more improvement on the mobility subscale of the low luminance questionnaire, which we had also developed. And so there was actually some behavioral realization that maybe their vision was better after the 30 days of high-dose vitamin A. So returning also to the patient-reported outcomes, I think it's important to look at the relationship between psychophysics and patient-reported outcomes. And you can actually see from our low luminance questionnaire, and also comparing it to the NEI VFQ, which is all the items asking about difficulty are focused on mostly bright light conditions. So what we found is the symptoms in older adults and those with early AMD are worse in dim environments, even for older adults in normal macular health. And the low light symptoms were more likely, if we followed people over time, were more likely to exacerbate over time than daylight vision. So as a result of this, pharmaceutical trials actually use our low luminance questionnaire as a patient-reported outcome, because FDA sometimes definitely likes to find out what patients think, not just how they see or what their retinal images are like. So about this time, my student, Greg Jackson, developed an alternative measure. Keep in mind, we were always using the slope of the second component to measure the rate of dark adaptation. So what he did was he developed something called the rod intercept time. And so here you can see cone recovery, quite rapid. Here we have normal adults with rod-mediated dark adaptation. And here you see the people with early AMD, much slower. And basically, rod intercept time is the duration required for sensitivity to recover to a criterion sensitivity value, which is in the latter half of the second component of rod recovery. So if you drop down, you can actually get a measure of rod intercept time. And the idea is that a higher number means slower recovery, a lower number means faster recovery. And so in the rest of my research, we're not really using the slope of the second component. We're going to be using the rod intercept time, and actually other people who are working in the field are using the rod intercept time as well. So at this point, we embarked on something we call the All-Star study, which is focused on early age-related macular degeneration. It is a natural history study on transitioning from normal to early AMD. And so the vast majority of the patients we enrolled at baseline were in normal macular health. And what we were interested in is, could we document that a certain percentage of people in normal macular health actually had delayed dark adaptation? And we wondered if that was a risk factor for basically that increased their risk for incident early AMD, for example, 3 years later. It was the design of the study. And so we actually embarked on this. And so we found that about a quarter of the people in normal macular health using the AREDS-9 classification system for how we define good macular health had delayed dark adaptation. And then what we found is that eyes with abnormal dark adaptation who were in normal macular health were 2 times more likely to develop incident AMD within 3 years. So basically, delayed rod-mediated dark adaptation is really the first identified functional biomarker for incident early AMD. This was surprising to many people. And in fact, we compared it to many of the usual outcomes that are used in AMD trials. For example, acuity, low luminance deficit, which is just a measure of mesopic acuity under low luminance conditions, contrast sensitivity for letters, and so on. But the cone-mediated vision was not associated with incident AMD. Mesopic acuity, which actually depends on rods and cones, was somewhat related, but not as strongly related as dark adaptation. We also went on in the first All-Star study to get blood specimens, and we found that 2 of the strongest genetic associations for AMD are associated with delayed rod-mediated dark adaptation. So in the column on your left, even before the AMD clinical phenotype emerges, if you look at the top plot, these people are in normal macular health. You see that these individuals with the ARMS2 polymorphism have delayed dark adaptation. That is, on the y-axis, they have elevated rod intercept time, if you look at this. And also, we found the same association in individuals with early AMD. What's interesting, we also found that association for CFH was associated with higher rod intercept time and those with early AMD. And then we looked at those with larger number of high-risk ARMS2 or CFH alleles. And if you look across here, you see it's going up. And actually, we only had 1 person with 4 high-risk alleles, and they had one of the worst dark adaptation in terms of the slowest dark adaptation rate. So this was very interesting to us, and we thought, well, at least we're looking at dark adaptation with respect to some of the genetic risk factors as well. We also looked at how these genotypes related to cone vision. And as you can see here, here's just one example. It's very flat. I could show you all the measures of cone vision, and it's very flat as opposed to here where it's going up with respect to rod intercept time. This was not a effect that was specific to all visual functions. It was something unique about dark adaptation. We then went on to, after we finished the first All-Star study, and the whole basis of that was to identify this first functional biomarker in slow dark adaptation, we went on to do another study called All-Star 2. And here we're just not focused on people in normal macular health. We actually focused we included those, but we also have groups with early AMD and intermediate AMD. And our purpose is to identify functionally validated structural endpoints. And so why would we bother to do this? Well, the time course and severity and types of vision impairment can actually reveal clues to the mechanisms of disease initiation. And we also thought we might be able to uncover some endpoints or outcomes that could be used in future research. So where are we? Oh, let me just, before I describe the structure of the study, we basically, Christine and I developed a conceptual framework for how visual function changes over time as it's tied to structure. So down here, we have people in normal macular health, early AMD, and these early structural changes have to do with changes in the choriocapillaris, Brooks membrane, the RPE, also drusen and subretinal SDD. And what we believe is that the very first functional sign, like I just showed you from our data, is compromised retinoid resupply from the choroid to outer retinal cells. But as the disease progresses, you start to see photoreceptor degeneration, and then you're going to probably see impairments in scotopic sensitivity until further in the disease, this neural circuitry is actually becomes corrupted, and then you see deficits in pattern vision, for example, acuity, letter contrast sensitivity. So this was kind of our general framework for thinking about the disease. So the design of the study is basically, we've finished the baseline study. It has about 500 people in the study. And then right now we're actually seeing them at the 3-year follow-up, but I don't have those data for you yet because we're still collecting them. So what did we do? Basically, they're people who age 60 and over. We recruited them from our clinics. They were in normal macular health, but they also had early AMD and intermediate AMD per the AREDS 9-step system based on color fundus photos. We measured many visual functions because we're trying to track all these over time to see what's the most promising. And you can see various aspects of acuity under photopic and mesopic conditions, light sensitivity using microperimetry under 3 different conditions: photopic, mesopic, and scotopic, and of course, rod-mediated dark adaptation. We also did extensive retinal imaging from fundus photos to OCT, OCTA, so that working with not only Christine, but also working with Fasada and Joel Hugh to try to tie some of the visual functional changes to the retinal changes. So far we've analyzed the baseline visual function data, and the purpose here in focusing on this is to inform what visual function endpoints would be the most useful in studies focused on the transition from aging to early and intermediate AMD. The analyses on the OCT and the OCTA variables are still ongoing, and you'll see some of those papers coming out in the near future. So one of the things that has been established with 3 different studies, one from our group, this is from the NEI group, shows that subretinal drusenoid deposits or reticular pseudodrusen actually accentuate the delays in dark adaptation as compared to people who have drusen only. And as you can see it here, this is the RPD group, the dotted line, and you can see the delay 40 minutes later, they're still recovering sensitivity. So whenever in All-Star 2, we wanted to make sure that we could identify those people with SDD. And so Christine Curcio's graduate student, Deepayan Carr, actually developed a multimodal imaging criteria system for evaluating from the near infrared reflectance, the UNFES OCT, and the B-scan OCT. And basically, he went through all 500 of the participants. And as you can see here, there was strong agreement between the 2 graders. It was almost 90%. So we have a reliable and valid way for identifying SDD from the OCTs. So the first thing we wanted to do in this study is look at all the visual functions. And as you can see, they're all listed here. Oops, sorry. Oops. And the very first thing I'd like to mention is that all the visual functions are worse. All the visual functions are worse when you compare normal to intermediate AMD. But what's really interesting, Yet if the focus is on preventing AMD, the only visual function that was worse when you compare early AMD to normal AMD was rod-mediated dark adaptation. And again, it speaks to the potential power of dark adaptation for being an endpoint in early disease. However, what we really wanted to do, all these visual functions have different metrics. So we wanted to basically standardize them so we could actually look at their distributional characteristics to see which ones are more prominent. So basically what we did was we standardized the visual functions with z-scores so that we could actually compare them. And we standardized them in such a way is that higher numbers indicate worse vision. And so this is the difference in the z-score from normal to intermediate AMD. And as you can see here at the top, all of them are listed here, but dark adaptation clearly has the largest z-score range from normal to intermediate AMD. So if what you're trying to do is, for example, a prospective trial that's tracking people over time, you're more likely going to be seeing more change in dark adaptation than you are in these other measures. Some of them have less than half the range that dark adaptation does. We also wanted to look at the distributional properties as a function of disease severity from normal to early to intermediate. So again, we used the z-score to standardize everybody. And we also computed probability density functions. And so what are these? And I'll try to explain this. It describes the likelihood of obtaining possible values for a continuous variable, and vision is a continuous variable when you're measuring it psychophysically. So it actually facilitates seeing the probability that certain values are observed per group. And it's kind of similar to a frequency histogram for ordered categorical variables, except in our case, we're dealing with a continuous variable. So to give you an idea of what one of these distribution looks like as a function of disease severity, this is the one for dark adaptation. And as you can see, the pink area are the normal subjects, green, early AMD, blue, intermediate AMD. And as you can see, there's a good separability between normal and intermediate AMD. The early AMDs are slightly worse. Keep in mind, higher numbers mean worse vision. But here you can see this, there's a rather prominent blue area indicating how intermediate AMD, the rate of dark adaptation there is much worse than normal. So it's interesting to compare dark adaptation to all the other visual functions we also measured. So here's the same graph of dark adaptation on the top left. You can see mesopic contrast sensitivity, photopic acuity, photopic light sensitivity measured throughout the macula. And as you can see, the blue area here for intermediate AMD is dramatically greater. In the case of dark adaptation, you see, like I said in the very first slide, you see a separation among these three groups, normals to intermediates, but it's not as dramatic as you see in dark adaptation. Here's another one showing mesopic light sensitivity and also scotopic light sensitivity. And I'm sorry, the plot is missing for the other one. But you see that dark adaptation is far surpasses what you see with the other visual functions in terms of understanding the separability of how visual function exists in these three groups. A question we were very interested in is, how does SDD operate in All-Star 2? So basically what we looked at is we've separated people into no SDD. These are people with drusen only, or in the case of normal macular health, no drusen. And then people who have SDD. And you can see SDD in normal macular health. It's a much lesser extent, but it is on occasion present. And as you can see here, in early and intermediate AMD, rod intercept time is much more increased. If you look at the gray, much more increased than if you compare it, compare it to the pink area. And the point I want to kind of emphasize here is I go to meetings and people say, well, you only see delayed dark adaptation in cases of where SDD is present, reticular pseudodrusen are present. But this shows you here that basically you see it in drusen only, it's just not as dramatic. So if we look at the probability density functions for no SDD and SDD, you can still see this prominent blue portion here. For here it is in SDD, it's more prominent there, but you still see it in no SDD. So it's important to realize that dark adaptation as an endpoint is not just isolated for cases of SDD. It can be used more broadly, even when only drusen are present. So finally, the question is, if you're measuring rod-mediated dark adaptation, obviously you're not doing this in the fovea. How far out should you go? And it's interesting, we compared measuring dark adaptation at 5 degrees to 12 degrees. 5 degrees is on the left. And as you can see that measuring it at 5 degrees separates aging from early and intermediate AMD much better than measuring dark adaptation at 12 degrees. So why would we measure it at 12 degrees? Well, that's the area where rod density is the highest, even in older adults. But measuring at 5 degrees, that's where rod loss is in aging and AMD is the largest. So it's kind of moving it near the fovea, where we know the fovea is a very threatened part of the disease, but it makes sense for us to measure at 5 degrees, because as you can see here, these effects are much larger, more dramatic than if you're measuring it out at 12 degrees. I think many of you have probably heard of the MacuStar study ongoing in Europe. They're measuring it at 12 degrees, and I think that's going to be a problem for them in actually learning much about dark adaptation in the MacuStar study. So to summarize, so research on AMD, at least from my perspective, should start to focus on preventing AMD and arresting its earliest progression, kind of like cardiovascular disease and heart disease, where we start intervening. Our studies have focused on that so we can prevent the end-stage disease. And so I think this is kind of an important area to emphasize. Recently, I was at a meeting on dry AMD. It was organized by pharmaceutical companies in Boston, but Foundation Fighting Blindness was there. And they began their presentation by actually making this recommendation that we need to start focusing on the aging to early AMD transition rather than focusing only on end-stage disease so that we can actually prevent the disease. So that was really interesting to hear that coming from the foundation. So the macula is rod dominant, and thus rods cannot be ignored in the study of how aging of the retina transitions to AMD. And rod loss, not cone loss, is characteristic of aging in early AMD. Both scotopic sensitivity and dark adaptation are impaired in these conditions. And what's really fascinating is that the patient reports align with the psychophysical findings. The first identified functional biomarker for incident AMD is delayed dark adaptation and not other types of visual deficits. And a mechanistic explanation that we like to use is that it's compromising metabolic exchange between the choroid and photoreceptors in AMD. Two of the strongest genetic associations for AMD are associated with delayed dark adaptation and not associated with cone dysfunction. And finally, the distributional properties of rod-mediated dark adaptation far surpass those of other visual functions, with a clear separation of the probability density functions that I showed you for normal aging when compared to early and intermediate AMD. In All-Star 2, we are now trying to tie, at least at the baseline data, the relationships between retinal structure and visual function. And we're also this past October, we began our 3-year follow-up because so many of the questions that we have really aren't cross-sectional questions like I've presented to you today, but are prospective questions and looking at changes over time. And I thank you very much for your attention.
few questions. Let's start with Bruce.
Good morning. Wonderful, wonderful talk. Thank you so much. My question is, I had it written down here, so I better just read it. But what role do rod mitochondria have in the transition from aging to AMD? And are the rod mitochondrial dysfunction worse in STD than in AMD?
Well, that's an excellent question. I'm not a biologist, so I haven't really studied that, but that's something that I know my partner in this all-star saga, Christine Curcio, is very interested in. It could be that the mitochondrial dysfunction, it could be contributing to the deficit. But from my perspective, I just don't know.
Thank you.
Okay, come up.
Hello, can you hear me?
Yes.
Yes.
Oh yeah, oh, nice presentation. I have one, kind of confusion or even a question. So the dark adaptation seems very, like it is very significant than other contrast sensitivity or virtual Barnier acuity, right? But isn't contrast sensitivity also related to the dark adaptation if you use a scotopic measurement? Right? Aren't they related? They're supposed to be linearly.
Well, keep in mind, okay, let me just make a distinction here. Rod-mediated dark adaptation is pretty much a retinally based phenomenon. The basic biology from Lamb and Pugh and other people who've worked on it really show that it's a retinal phenomenon. Contrast sensitivity really depends on the entire visual system. It's heavily cortically influenced as well as obviously retinally influenced. So you can measure contrast sensitivity under scotopic conditions, but it's extremely difficult to do so because it's just hard to see. So
I see.
So
Otherwise, yeah, I mean, usually contrast sensitivity is contribution from both rod and cone, right? That's what you're saying.
well, it could. I mean, it depends under what light levels you're studying under. If you're studying it under mesopic conditions, both rod and cones would be operative. Photopic conditions, cone. But it's very difficult to measure contrast sensitivity under scotopic conditions, at least in humans.
I see. All right. Thank you.
Cynthia, if I may have one academic question and one real question. So my academic question to you, I don't know who coined the term dark adaptation, but we don't dark adapting, we're recovering sensitivity. And so light adaptation, of course, we're adjusting to background light, but dark adaptation, it's not a very precise term, is it? What is your thought on that? Or it is what it is?
I think it may go back to the days of Helmholtz. He probably studied it at some point. It's always been called dark adaptation all the way through the 20th century. They were studying, vitamin A deprivation, etc. So it is a funny term. It's really the recovery of sensitivity after a bleaching light. So.
And the real question is, at the end of your presentation, which by the way was one of the clearest I have ever seen, the STD, you speculated that maybe flow of retinoids from RPE to the retina because of SDD is their presence, some barrier. But then you show that this dark adaptation, let's use that term, is affected also in patients which do not have this barrier. So what are the other mechanisms that it comes to your mind could be responsible for this slowing down recovery of sensitivity?
Well, I think drusen and basal-linear deposits, the oil spill, as Christine calls it, that's happening in the retina, those drusen and those sorts of deposits could also slow dark adaptation as well. I think one of the, let me just mention, one of the interesting things is SDD are more prominent further out in the retina. They seem to have a heavy influence on what's happening in the central retina. So exactly what's happening with SDD is kind of what the mechanisms are in terms of understanding dark adaptation it remains a mystery. Anyway, that's how I would respond to your question.
But this is on the basal, drusen on the basal side, so shouldn't have any effect on the visual cycle. At least not in the immediate, like acute phase when you're exposed to light, right? It may be long term, yes, but not in acute space.
Right, right.
Okay, let's move to James.
Okay, so Chris kind of asked my question already, but I'll modify it a bit. So in terms of molecular mechanisms, you mentioned the genes that you identified, which are associated with AMD that correlated with delayed dark adaptation. I was wondering if there were any genes that are known to be associated with AMD that did not correlate with delayed dark adaptation.
So we haven't done those studies yet. Some of the lipid related genes would be good candidates for investigation. I know, Christine, we actually on All-Star 2, we have DNA as well, and we're trying to get whole genome sequencing to also look at these other genes as well, and we're still trying to raise the money to do that. But I think the lipid genes would also be relevant as well.
Francesca.
So my question should have come before Chris's, but I will ask it anyway. So it is fascinating that this subretinal pseudodrusen deposit correlate with the delayed dark adaptation. So you showed us the OCT images. Did you have any chance to use SLO to somehow correlate the density of this subretinal drusen deposits with the delayed dark adaptation?
That's what we're doing now. We're trying to tie the delayed dark adaptation more precisely to the retinal imaging, but that's a very big part of the project.
Okay, Barb.
Thank you. Hey, Cynthia. Pleasure seeing you again. Quick question. So a bunch of years ago, I think it was both Schmetterer as well as Steve Moss that have showed that in CFH risk variants that you have a reduction in blood flow, and that is something that actually occurs prior to AMD onset. And so I was wondering whether there's a correlation between sort of the timing that you first have a reduction in blood flow and then this reduced rot intercept time that you suggest is in part due to loss of vitamin A delivery. And in the same context, has anybody actually looked at C-wave analysis, which is a measure of like RPE health? And if there's really a reduction in glucose transport to the RPE, which we know creates a metabolic crisis for retina and RPE in AMD, whether there's a correlation in C-wave amplitudes with anything that with the rod measures that you're seeing.
I don't know of anyone who's looked at the first thing or the second thing you mentioned. I think those would be great mechanistic hypotheses to explore. I'm wondering how. I guess I'm unfamiliar with the techniques, but how would you do that in humans? I guess I turn the question back to you.
So Schmetterer did all of his work in humans. Moss and Jeffries did theirs in mouse. And C waves, they can be recorded in both. I mean, it's just another measure of in ERGs.
I see. Okay.
The sort of the delayed. I mean, the longer response. It's a long light stimulus, and I forgot how long it lasts in human, but in the mouse retina we see it with a 2-second stimulus.
Yeah, I mean, I think those are great hypotheses and they should be followed up on. I think
and that sort of indirectly also gets at Bruce's question on mitochondrial contributions.
Exactly. Yeah. But I think that would be a great way to go. I think one of the challenges in these big studies, keep in mind, we're studying 500 people and we have to be so prudent. I mean, the protocol is like 4 hours long and then they come in twice, 3 years apart. But I think these sort of mechanistic questions could be, if there were an all-star 3, they could be added and probably would serve as a much better, it would lead us to a much better understanding of for why dark adaptation may be delayed. It may not just be the vitamin A nutritional deficiency.
Michael.
Thank you.
Let me turn on my camera. Yeah, I think this is really important because delayed dark adaptation is a really serious precursor to AMD. So I think it is quite quite important that we sort of get to the bottom of it. But my question was kind of following up on what Chris asked earlier, and your experiments where you showed that increasing vitamin A helped. But I would ask whether it's possible that it's kind of more basic than that. That it's that the visual cycle is somehow suppressed, and that is kind of more immediately affecting rod recovery. Have you thought about that?
No, but I think that's an excellent possibility that could be explored. Yeah, I mean, I would agree with you that it could be the visual cycle itself rather than just the vitamin A.
Yeah, we've done studies with mouse where visual cycle is suppressed due to RP65 mutations, and we see delayed rod recovery, but it's obviously not an AMD model, but it could be along the same lines. Thanks
Yeah,
very much.
I would agree with you. Yeah, I think that would be a great avenue.
So adding this to Michael's comments and question, wouldn't you expect in RP65 heterozygotes that those patients will develop AMD more frequently or severely? Michael?
Yeah, that's something that has always been in the back of my mind, that patients who are heterozygous, like you mentioned, carriers, in essence, might in later stages, later in life, have delayed dark adaptation, that it might not be clinically visible when they're younger, but possibly when they're older. So that I think that's something that if given infinite resources, it should be checked to see if there is correlation.
Okay, so we have infinite resources in NIH. We'll be done. John.
Hello. Thank you for this great presentation. I was wondering if this slower rod-mediated dark adaptation is observable before any structural features can be observed by imaging, or are there any subretinal features? I mean, are there any structural features beyond just the subretinal drusenoid deposits that precede this delayed dark adaptation?
That's an excellent question. I think what the first All-Star study showed is that if you use the traditional or the conventional imaging technique of fundus photography, the dark adaptation deficit does precede anything you would see in the fundus. But with respect to OCT, we're now going through all the OCT data to see if there's something about the people who are in normal macular health according to conventional clinical criteria, whether their delayed dark adaptation is tied to any specific OCT features. And so that's an excellent question, and that's a big part of what we're trying to do with the baseline data here.
Is that it?
Hello, Dr. Owsley. Thank you for your talk. I wanted to ask about whether rod-mediated dark adaptation could be used as an early marker of visual function decline, especially to screen people who need to work at night, such as policemen, military servicemen, or truck drivers, especially in older individuals.
Yeah, that's interesting. You know, the military, I think it was during World War II, studied, and also in the early 50s, studied a lot of, studied dark adaptation in particularly in the Army, I think. And it, you know, it could be that there are individual differences. In fact, in the military, if you go back to some of the very old articles, there's quite a bit of individual variability, even among people who are considered normal, whatever that is. So I think dark adaptation may be useful for understanding individual differences, but keep in mind in individuals in normal macular health, I mean, there are, even in young adults, we see variations. So it could be something worth looking at in terms of individual differences and sort of certain in terms of occupational issues. Would this person have better night vision? You know, you could probably document it with dark adaptation techniques.
Raphael?
Hello, Dr. Owsley. Thank you for fascinating talk. I have a technical question related to patient compliance during the dark adaptation measurement. Is it very difficult to achieve? Because I think at some point you mentioned 40 minutes dark adaptation. So I was wondering what's the success rate of the measurements?
So you know, one of the reasons that retina clinicians who typically serve as the advisors to pharmaceutical companies in terms of their study designs, one of the major problems is the length of the test. And so, you know, in some individuals, we can go as long as 40 to 45 minutes, and they're still not, they still haven't reached the rod intercept time. So what we're trying to do is find ways to shorten the test, possibly reduce the bleach, the strength of the bleach, or we're also looking at some machine learning possible solutions to shorten the test, because the major problem, I mean, in talking to the pharmaceutical companies, they're very, very excited about this test, but when they get their study advisory boards together, the retina clinician scientists say there's no way I'm going to have a test that takes maybe even 20 minutes to measure. And so one of the things that I'm trying to figure out is how we can shorten this test in such a way that we'll still have the sensitivity potentially, or the effect sizes to be useful in a trial, but try to shorten it so that it would be more palatable to retina specialists who advise the pharmaceutical companies. I mean, when I go to meetings like the Ryan meeting or any of the AMD type meetings, if it's primarily retina specialists, they just say, oh, there's just no way I can use this test because it's too long. My only comeback is that, for example, you're probably all familiar with the Sparks Therapeutic trial. They used a low luminance mobility course that took at least a half hour, 45 minutes. In inherited retinal degenerations, there's ERG protocols that are not brief. So I think for the purpose of showing that a treatment helps, I think we need to get over a little bit the time problem.
Thank you so much.
All right, we have two more questions and then we go to final comments by our famous Dr. Andrew. So we will start with John and then Chris.
Hi, just a quick question. Does anybody know what materials or make up the SDDs?
Yeah, Christine Curcio has done a lot of research on what they are. They're definitely contain lipids. They're not identical to drusen, but they're similar to drusen. any of her work would explain better what it actually contains.
Any fragments of like shed distal tips of outer segments?
I don't know. That's a good question. I personally don't know.
Okay.
Possible. It's possible. All right, so let's move to Chris.
Yes, that was a fascinating talk. I have a quick question, sort of following up on what another question. Are there any ethnic or racial differences in the dark adaptation? Certain populations have, are more effective, or is that going to tie back to mitochondria, I guess?
Yeah, excellent question. So in our studies here, because of our exclusion criteria, our samples here, the All-Star studies, and I would also say this is true for some of the other groups around the country that are studying it, most of the participants are white of European backgrounds. For example, here in our, where we recruit our patients from in our clinics, we exclude people with other eye conditions, for example, glaucoma, diabetic retinopathy. And of course, in our clinics here, we have a high percentage of African Americans, of course. We live in Birmingham, Alabama, but those diseases are more prevalent, so they don't end up being in our study samples. All the studies I know of that are larger, kind of more epidemiological in nature, mostly focus on white people. So I can't answer your question, but I think it's excellent.
Thank you very much.
Okay, and now I will give you Andrew.
Thanks again for a really interesting and inspiring talk. I think that one of the things that struck me about your talk was, when you showed the normals versus early intermediates, that there are these kind of separation of populations that have some people that have dark adaptation elongation, some people that really don't, with either pseudodrusen or no pseudodrusen. That kind of speaks to the multifactorial nature of this problem, and maybe it comes down to choroidal blood supply in addition to diffusion barriers, in addition to even the blood supply in the choroid regulating local heat in the retina during visual tasks. But my real question I wanted to kind of ask you about comes to the dark adaptometry test, where you flash a light and then you have a microperimetric approach, I believe, to looking at 5 degrees or 12 degrees. Is that true? It's the subject responds whether they can see a light flashing at that location?
Yes, and it's not really microperimetry because it's not fundus controlled.
Okay, and have you or has anybody ever tried to use multifocal ERG in a similar kind of protocol, and then look at the recovery of the ERG waves in multiple places at the same time?
No, we did do multifocal ERGs back in the 90s, and we also did scotopic ERGs, and we didn't find it was so sensitive. It wasn't sensitive to disease severity because forget the multifocal for a minute, because that was under cone conditions, but the scotopic ERGs, it's a mass response and they're getting a whole flash all over the retina. So it just didn't differentiate among different stages of AMD very usefully. Pharmaceutical companies have been interested in using ERGs, but with our results and other people's results showing that you can't really differentiate among different levels of disease severity, it was sort of abandoned, so to speak.
Okay, so no one's really looked at the multifocal ERG kind of sensitivity recovery after a white flash? No.
Keep in mind, multifocal ERGs are common in the sense that you can do them under cone conditions, photopic conditions, but it's very difficult to do multifocal ERGs under scotopic conditions because of all the stray light problem. And so Don Hood actually published a paper on this, how difficult it was. So people haven't really followed that route after that.
Thank you.
Cynthia, I wanted to thank you again for participation today. It was a wonderful conversation. So thank you very much.
Thank you. Thank you very much.